Protoplast culture. Protoplasts are naked plant cells without the cell wall, but they possess plasma membrane and all other cellular components. They represent the functional plant cells, but for the lack of the barrier cell wall, protoplast of different species can be fused to generate a hybrid, and this process is referred to as somatic hybridization or protoplast fusion. Sy- Hybridization is the phenomena of fusion of a normal protoplast with an enucleated without nucleus protoplast is the protoplast that results in the formation of a hybrid or cytoplasmic hybrid. Protoplast isolation refers to the separation of protoplast from plant tissue. Important to isolate viable and uninjured protoplast as gently and as quickly as possible involves two methods: mechanical enzymatic mechanical method. Tissue is immersed in 1 molar sucrose until protoplasm shrunk away from their enclosing cell wall. Plasmolysis. Plasmolysed tissue is cut with a sharp knife and at such a thickness that only cell walls are cut. Plasmolysed cell. Undamaged protoplasts in strips are released by somatic swelling when placed in a low concentration of sucrose solution. Problem encountered: some cells release uncut complete protoplast while the rest produces broken dead protoplast. Enzymatic method refers to the use of enzymes to dissolve the cell wall for releasing protoplast. Involves two methods: direct method, one step only; sequential method, two-step method. Direct method: incubation of leaf segments overnight in enzyme solution. Mixture is filtered and centrifuged. Protoplast forms pellet, then washed with sorbitol and re-centrifuged. Clean protoplast float. They are pipetted out. Sequential method: two enzyme mixtures, mixture A and mixture B, are used one after the other. Leaf segments with mixture A, meso, uh, meserozyme in manifold at pH 5.8 are vacuumed infiltrated for 5 minutes. Transferred to a water bath at 25 degrees centigrade and subjected to slow shaking. The enzyme mixture is then replaced by fresh enzyme mixture A and leaf segments are incubated for another hour. The mixture is filtered using nylon mesh and centrifuged for 1 minute. Washed 3 times with 13% methanol with 13% mannitol. Cells are then incubated with enzyme mixture B. Cellulase in mannitol solution at pH 5.4 for above 90 minutes at 30 degrees centigrade. The mixture is centrifuged for 1 minute so that protoplasm form a pellet and clean 3 times with sorbitol. Purification of protoplast. Purification of protoplast. Protoplasts are purified by removing undigested material, debris, bursts, protoplast, enzymes. Debris are removed by filtering the preparation through a nylon mesh Enzymes are removed by centrifugation, whereby the protoplasm settle to the bottom of the tube and the supernatant removed with the help of a pipette. Intact protoplasts are separated from broken protoplasts through centrifugation and removed by a pipette as they are collected at the top of the tube. Isolated protoplast culture. Isolated protoplast can be cultured in an appropriate medium to reform cell wall and generate callus. Optimal culture conditions, optimal density to the culture, optimal oxygen to cytokinin ratio, glucose and sucrose maintain osmoprotectant in the medium temperature 20 to 28 degrees centigrade, pH 5 to 5.5 to 5.9, 0.25% casein hydrolysate, BAP and NAA. Culture of protoplast. Protoplast cultured in a suitable nutrient media first generate a new cell wall. The formation of a complete cell with a wall is followed by an increase in size, number of cell organelles and induction of cell division. The first cell division may occur within 2 to 7 days of culture, resulting in small clumps of cell, also known as microcolony, within 1 to 3 weeks. From such chumps, there are two routes to generate a complete plant, depending on the species. Plants are regenerated through organogenesis from callus masses. The microcalli can be made to develop into somatic embryos, which are then converted into whole plant through germination. Importance of protoplast culture. The protoplast in culture can be regenerated into a whole plant. Hybrids can be developed from protoplast fusion. It is easy to perform single cell cloning with protoplast. Genetic transformations can be achieved through genetic engineering of protoplast DNA. Protoplasts are excellent materials for ultrastructural studies. Isolation of cell organelles and chromosomes is easy from protoplasts. Protoplasts are useful for membrane studies, transport and uptake processes. Isolation of mutants from protoplast cultures is easy.